Okay, so this is the first agent of the anthracycline antibiotics. That is, the name of the drug is the doxorubicin hydrochloride. Okay, doxorubicin hydrochloride. Now, the C13 substituent of the doxorubicin is the hydroxymethyl. So, this doxorubicin drug, it is having a hydroxymethyl group at carbon number 13, and which retard the action of the cytosolic aldoketo-reductase and slow down the conversion of the less active and chronically cardiotoxic doxorubicinol. So this enzyme, it inhibit the action of the aldoketo-reductase. Means what? That is, the parent doxorubicin structure is more active as compared to its metabolic alcoholic compound. This contribute to the longer duration of action compared to the analogs that have CH3 at this position. So means what? That is, the Daunorubicin drug. It contains the CH3 group at this position. Okay, it contains the Daunorubicin drug. It contains CH3 group at this position, and the duration of action, and therefore the duration of the action of Daunorubicin is less as compared to the Doxorubicin. Doxorubicin is highly lipophilic and concentrate in the liver, lymph nodes, muscle, bone marrow, fat, and the skin. The doxorubicin, it is either used alone or in combination therapy to treat a wide range of neoplastic disorders, including hematologic cancers and solid tumors in breast, ovary, stomach, bladder, and the thyroid gland. So that is all about the doxorubicin hydrochloride. Now the next drug is the daunorubicin. Now check out over here. In the previous structure it was hydroxymethyl. Now in this structure it is a methyl group at the 13th position. As the remaining part is same, the structure is same. There is no any difference. So the absence of the hydroxy group at C14. So this one is the 13th carbon, and this one is the 14th carbon. So there is no any hydroxy group at the 14th carbon, and hence the absence of the hydroxy group at C14 in daunorubicin result in a faster conversion to less active metabolite, that is the daunorubicinol. So what we discussed in the earlier slide, that the previous drug inhibit the enzyme aldoketo-reductase, and thereby they slow down the conversion to its less active metabolite. But because of the absence of this hydroxy group, the daunorubicin drug fastly get converted into the less active metabolite, that is the daunorubicinol. Now, this agent is given by the intravenous route for the treatment of the acute non-lymphocytic and the lymphocytic leukemia. The major difference between these two agents being the spectrum of cancer that they are used to treat. So, if we check the activity of both the compound, that is the daunorubicin and the doxorubicin, so the doxorubicin is more potent as compared to the daunorubicin, right? Why it is? Because of the presence of the hydroxy group in the doxorubicin, which inhibit the enzyme aldoketo-reductase, and thereby the conversion into the less active metabolite is they slow down the conversion into the less active metabolite. So that is what the difference between the doxorubicin and the daunorubicin. So this is what all about the anthracycline antibiotics. Now the next one is the actinomycins. Now actinomycins. What is actinomycins? If you call it as an actinomycin, then you have to recall the name of the drug, that is a bleomycin. Yes. So what I have discussed, that if you call the name of the class actinomycin, then at that time you have to recall the name of the drug, that is the bleomycin. There is one agent in this actinomycin class, and the name of the agent is the bleomycin. So actinomycin, They are the group of compound which is isolated from the various species of the streptomyces, and all these compounds they contain phenoxazone chromophore, but which differ in the attached peptide chain. So this is the structure of actinomycin. Okay. Now, if you check out the structure of this actinomycin, now the actinomycin is not actinomycin is the name of the class. Actinomycin is the name of the class, and this is the structure of the dactinomycin or the actinomycin D, right? Dactinomycin or actinomycin D. So this is the structure of the dactinomycin. Now, if you check the structure, so this structure contains the phenoxazone ring, phenoxazone chromophore, which is attached to the two peptide chains. These peptide chains are attached at the first position and at the ninth position of the phenoxazone ring. So check out properly the structure. Now the mechanism of action again of these actinomycins is same as discussed with the earlier class, that is the anthracycline antibiotic. So what actually the mechanism of action of all these drugs? That is, they contain these tricyclic aromatic ring, and this tricyclic aromatic ring get inserted into the DNA base pairs. They get inserted into the DNA base pair, and after get inserting into the DNA base pair, they inhibit the DNA replication process. Whereas these peptide chains, they also bind to the minor groove of the DNA, and they participate in the inhibition of the DNA replication process. So from this group emerged actinomycin D, which is known as the dactinomycin, and they contain identical pentapeptides. So if you check out, they contain the identical pentapeptides. See, it contains the threonine, they contain the valine, they contain the proline, they contain the SAR amino acid, then they contain the mevalinic acid. So both the pentapeptides are same. So this drug they contain the identical pentapeptides which is bound through an amide linkage. So these pentapeptides are bound through an amide linkage. How they bind through the amide linkage? That is L-threonine, as these are the amino acids. So amino acids contain what? The amine group and the carboxyl group. So the amine group of this L-threonine with the carboxyl group of the phenoxazone ring. So by this amide linkage, that is C-O of this phenoxazine and the nitrogen of this threonine, as these are the amino acids, so they contain the nitrogen. So by these amide linkage, the phenoxazone ring get attached to this pentapeptide chain. Okay. Now, which pentapeptides they contain? So they contain the L-threonine, D-valine, L-proline, then the sarcosine, and the L-methylvaline, and they form a lactone ring, and they interact with each other, and they form a lactone ring. Now, this dactinomycin drug, as we have discussed, that is, they bind non-covalently to the double-stranded DNA by intercalating between the guanine cytosine base pair, and which inhibit the normal DNA function. The structural feature of dactinomycin important for its mechanism of the cytotoxicity is the planar phenoxazone ring, which facilitate intercalation between the DNA base pair. So what we discussed earlier, that is the planar phenoxazone ring, it get intercalated between the guanine cytosine base pair of the DNA, and thereby they inhibit the normal functioning of the DNA. Whereas these peptide loops, they are also get inserted into the minor groove of the DNA. The preference for the guanine cytosine base pair is thought to be partly related to the formation of a hydrogen bond between the two amine group of guanine and the carbonyl of the L-threonine residues. So now in this slide and again in the next slide, they have mentioned the various interaction They have mentioned the various interaction which takes place between the drug and the DNA. Okay, so just check it out properly. Just listen me carefully. That which kind of interaction takes place between the drug and the DNA, and how they get intercalated between the guanine cytosine base pair. So first one is what the preference for the GC base pair is thought to be partly related to the formation of hydrogen bond between two amino group of guanine. Now guanine is present over here, that is guanine is present in the DNA. So the hydrogen bond formation takes place between the two amino group of guanine and this carbonyl group of the L-threonine residues, right? So by this interaction, that is the two amino group of guanine and the carbonyl group of the L-threonine, they interact with each other to form a hydrogen bond. So this is the one interaction between the drug and the DNA. Then the second one, additional hydrophobic interaction and hydrogen bonds are proposed to form between peptide loops and the sugars and base pair within the minor groove. So what this type of interaction? So this is the hydrophobic interaction. This is the hydrophobic interaction and the hydrogen bond is formed between the peptide loops. So these are the peptide loops, two peptide loops. The hydrogen bond is formed between the peptide loops and the sugars. Sugars over where, as we know, the DNA base pairs are made up of the nucleotides, and the nucleotides are made up of what? Nucleotides are made up of, yeah. So as we know, the nucleotides are made up of the base, either purine base or the pyrimidine base, then the sugar and the phosphate group. So the hydrogen bond is formed between the peptide loops. So these are the peptide loops and the sugar. Sugar of what? That is the nucleotide base and the base pair. Either it is purine base or the pyrimidine base within the minor group. Right? So the peptide loops, they form hydrogen bond with sugar, base pair, with sugar and the base pair. So that is what this is the another hydrophobic interaction. Then the primary effect of this interaction is the inhibition of DNA-directed RNA synthesis and especially RNA polymerase enzyme. DNA synthesis may also be inhibited, and this agent is cell cycle specific for the G1 phase and the S phase. So as you know, the primary mechanism of action of this agent to inhibit the DNA synthesis, and these agents are particularly cell cycle specific. They act on G1 phase and the S phase of the cancer cell cycle. So when sliding between adjacent DNA base pair, the actinomycin orient itself perpendicular to the main DNA axis, allow pentapeptide lactone units to bind to residue in the minor groove of DNA through hydrophobic and hydrogen bond. So when this phenoxazone ring get inserted between the guanine cytosine base pair, then at that time the drug orient itself perpendicular to the main DNA axis, so which allow the pentapeptide get bind to the minor groove of the DNA through the hydrophobic interaction and by forming the hydrogen bond. An affinity enhancing bond between the threonine carbonyl oxygen. So where is the threonine? Which is present at the peptide, right? Which is present at the peptide. So these peptide are made up of what? Amino acids. So all these are the amino acids. And if they are the amino acids, so they contain the two groups, that is the amine group and the carboxyl group. As we know, the amino acids, they contain the two functional group, amine and the carboxylic acid. So another interaction takes place, or the affinity enhancing bond between the threonyl carbonyl oxygen and a protonated C2 amino group of guanine also formed. So this is another interaction, that is the interaction between the threonyl carbonyl oxygen and the protonated C2 amino group of the guanine. So guanine present at the DNA. So this is one type of interaction. Other hydrophobic and pi stacking interaction form between the lactone and the DNA residues, particularly guanine and cytosine hydrogen bond. Of course, they form the interaction between with the guanine and cytosine of the DNA, because these agents get intercalated between the guanine and cytosine residues of the DNA. So all these actinomycins, they form a hydrogen bond or hydrophobic interaction with the guanine and cytosine residue of the DNA. The affinity enhancing bond between the threonine carbonyl oxygen and a protonated C2 amino group of guanine also form. Okay, it is again repeated, which is mentioned before in the previous paragraph. So it is again repeated. So just skip this paragraph. So the other okay, again it is repeated. Sorry, again it is repeated. The binding of dactylomycin to the DNA, although non-covalent, is much stronger than that observed with the anthracycline. So these binding of the dactylomycin or the actinomycin to the DNA, it is stronger if we compare the interaction of the anthracycline to the DNA. Drug dissociation from DNA is slow, leading to pseudo-irreversible effect. Dactylomycin, they block the gene transcription and the translation process, and again they inhibit the RNA polymerase enzyme, result in the decrease in the RNA synthesis and ultimately the protein synthesis, which is required for the synthesis of the DNA and the RNA. And these are the various uses of these dactylomycin. They are used in the various types of the cancers. So as I have discussed with you, if the name actinomycin come, so of course you have to recall the name of the drug, that is the bleomycin. Okay, bleomycin. Now bleomycin is a glycopeptide antibiotic complex isolated from the Streptomyces verticillus. Okay, bleomycin is a glycopeptide antibiotic complex which is isolated from the Streptomyces verticillus. At least 13 different fractions of the bleomycins are isolated, but out of which only A2 means there are there is a mixture of these bleomycin are isolated. One is the A2 and the B2 fraction. Okay, means they isolated a compound called as the blinoxane. Blinoxane. This blinoxane contain A2 fraction around 55 to 70% and B2 fraction around 25 to 32%. So out of which A2 fraction appear to possess the greatest antineoplastic activity. Through DNA intercalation, the aromatic bithiazole ring system positions bleomycin for the DNA destruction via cytotoxic free radicals. So now we will check out the structure of the bleomycin. Now this is the structure of the bleomycin. So as we have discussed, they contain this bithiazole ring system, and this bithiazole ring system get inserted into the DNA base pairs. So this bithiazole ring system get intercalated between the DNA base pair. As we have discussed in the actinomycin there was a ring called as a phenoxazone ring. So that phenoxazone ring get intercalated between the GC base pair. Now in this case, this bithiazole ring system get intercalated between the DNA base pair. Okay, wait for a few minutes, students. Bleomycin, uh, there are two bithiazole ring system which is get intercalated into the DNA, and thereby they inhibit the normal functioning of the DNA. Through DNA intercalation, the aromatic bithiazole ring system positions bleomycin for the DNA destruction via cytotoxic free radicals. So these agent after get inserting into the DNA base pair, they uh, generate some cytotoxic free radical which destruct the DNA. The disaccharide, polyamine, imidazole, and the pyrimidine ring are electron rich and readily chelate intracellular ferrous ion. So these all are the rings, the disaccharide ring, polyamine ring, imidazole ring, and the pyrimidine ring. So these all rings are present in the structure of the bleomycins. And these rings are the electron rich and they chelate the intracellular ferrous ion, which is get involved in the synthesis of the cytotoxic free radicals. Once ferrous ion get chelated, it is get oxidized to the ferric ion, with a concomitant reduction of the bound oxygen. The ferric hydroperoxide bleomycin complex is considered the cytotoxic form. So I just skip this slide. Directly, I will discuss with this mechanism. Now, if you check out, this is the structure of the bleomycin, which is mentioned over here in a short form, right? This is the structure of what? Bleomycin. And this is your DNA. This is what? The DNA. Now, what the bleomycin do? That bleomycin abstract a hydrogen ion from the DNA to form a radical. To form a radical. This radical, now check out over here. This radical then get reduced to the synthesis of the base propanol, and this base propanol then alkylate the protein at the cysteine residue. This base propanol then alkylate the protein, which is these proteins are necessary for the synthesis of the DNA. Okay, are you getting me? Means what was studied in the DNA alkylating agent. In that we have studied that is chlorambucil. Then again we have studied in nitrosoureas. That is these radicals which is synthesized in this process, the radical get reduced to the base propanol, and once base propanol is get synthesized, they alkylate the protein which is involved in the synthesis of the DNA. As we know, for the synthesis of the DNA, they also require some proteins. So they alkylate the proteins, and later on alkylated protein which get synthesized, they are not available for the synthesis of the DNA. So the DNA is get damaged because that DNA is not completely formed. The DNA is not completely formed. They require some proteins, but as the proteins are alkylated, so there is less availability of this protein for the synthesis of the DNA. So this is what about the mechanism of action of the bleomycin. Then again, the drug bleomycin is get destructed by the enzyme bleomycin hydrase, which is mentioned over here. Check out the name of the enzyme, bleomycin hydrase. So with the help of the bleomycin hydrase enzyme, the drug is get destructed, and thereby the tumor get resistant against the bleomycin agent. Conversely, tumor which are poor in the bleomycin hydrase enzyme respond well to this agent. Of course, if this enzyme is not present in the tumor, this enzyme, bleomycin hydrase enzyme, if it is not present in the tumor, then the drug show properly its antineoplastic action. And this bleomycin agent again used in the various types of the cancer.